All right, good morning, everybody. So my name is Rob Phillips, and this is the end of the line for me. So today I'm going to talk about what I consider to be, if not the greatest model in biology, at least one of them. And so to motivate that, I want to remind you of where I ended up yesterday. And where we ended up was we had talked about transcription factors and how they interact with DNA and how those interactions with DNA elicit different levels of regulatory response. But what we really had not included in our description at all was the idea of feedback. And another way of saying it is we hadn't included the idea that somehow, as Art has just told us, that there's all sorts of signals that can come and change the situation. In other words, you know, you have some hormone or any chemical that you might imagine, it will get into the cell and it can alter the behavior of transcription factors. So here I'm showing you a cartoon which is from the Alberts book, and all I really want you to take away from this is the idea that there's feedback and it's implemented by the product of this gene. It's very interesting. So let's say that you want to monitor the amount of tryptophan, an amino acid. And if the amount of tryptophan is high, then you want to turn off production of more tryptophan synthesis. But if you're lacking tryptophan, you want to make more tryptophan. So as you can see here, the repressor actually has a binding site in this cartoon for tryptophan. And when it's bound to the repressor binds to the DNA and shuts off the gene of interest. So really that's going to be the subject of today, not from the standpoint of transcription really, but much more broadly. So what classes of models can address this? And the thing that I'm going to be interested in, at least in the context of transcription, but more broadly, is response curves. So here I'm showing you three different induction curves, the idea being that as you put in the inducer, you get more and more gene expression. And what I want to think about is, you know, how would we think about such problems? So that leads me to today's topic. So we're right now celebrating the 50th anniversary of what Monod referred to as the second secret of life. So the idea is broadly known as allostery. So this is a 1963 paper by Monod, Change, and Jacob. It's called Allosteric Proteins and Cellular Control Systems. And then 2 years later, they wrote a paper entitled On the Nature of Allosteric Transitions, a Plausible Model, this one by Monod, Wyman, and Change. And this has come to be known as the MWC model. And that's really what I'm going to talk about. So, well, let me see what Monod has to say about it. He says the first paper was really on the idea of indirect regulation, which I think is the really important idea. The second paper is a physical-chemical interpretation of this fact in terms of the geometry of the molecule. And as of several months ago, the first of these papers had about 1,500 unique citations. This one had around 6,000 unique citations. And what I want to do is I want to entertain you in part, and I guess I also want to inspire you, if I can, with this model and its broad reach. So let's start with allostery. So a few months back in Paris, they had the 50th anniversary of the MWC model and allostery and these papers. There was a conference, and there were many distinguished speakers. And one of the things that I thought was very interesting about it is that all of these different topics came up. And so let's just review what you're looking at. So does anyone recognize this? I'll give you a hint. Yeah, hemoglobin. So there you go. This is lac repressor. This is a G protein coupled receptor. This is the ATP synthase. This is a molecular motor. This is the nicotinic acetylcholine receptor, and that's the ribosome. And it's fair to say, you know, if I took those, you know, 7 molecules, I could build a whole course around them, because I could say, well, you know, I'm going to talk about the central dogma, and I'm going to talk about signaling, and I'm going to talk about metabolism, and I'm going to talk about synthesis and transport. You know, it's really got all of biology. Not to mention, it's got the great storyteller of evolution, which is the ribosome. So allostery is this idea, and even though it might seem trivial at this point, it was very hard won. It was not an easy success story. Allostery is the idea that a molecule has two, let's for simplicity say it has two conformations, active and inactive, all right? And it's going back and forth between active and inactive all the time. But the thing that happens is that in the presence of a ligand, the free energy balance between those two states is shifted. So you might imagine that in the absence of ligand, this is the favored state, and maybe 99% of the time it's in this state with little tiny fluctuations now and then to this state. But now in the presence of some ligand, we shift exactly to the opposite scenario, and 99% of the time we're in this state. Okay? And so that's the idea of allostery. And what I like about this is that, and what I hope I'm going to show you, is that this, I also think, is an idea that really can serve as an all-encompassing biological concept. So here, what I wanted to do was, it's a little chance to talk about one of my favorite topics, which is whales, and just to remind you about the notion of phylogeny. So phylogeny is, at least in modern terms, a way of coming to terms with evolutionary relatedness. And you know, it's not very far away from here, but the vast majority of the fossils, the transitional forms for whales were found. I don't know, I'll take a minute just to tell you the story of Philip Gingerich. Some of you heard me say this before, but probably most of you haven't. So Philip Gingerich is a paleontologist, and his interest was, I think this should thrill you as Indians. He wanted to come to Asia, and he wanted to look at the gradient of fossils that had to do with the collision of India into Asia. So his scenario was there should be a bunch of animals that walked off of India into China. And so he would like to find the fossils. That was his plan. And as a young man, he kept coming to Pakistan, and each summer he would dig. And each summer he would find more whales. And so finally he just gave up. He said, all right, I'm going to give up my original dream, which is to watch the fossils and you know, it's still a great project. I've got another one in mind that's kind of similar in a funny way. So I spend a lot of time in Korea these days, and I think a fascinating place for doing a study of invasive biology and evolution would be the DMZ. So the demilitarized zone, because basically for 50 or 60 years it's been left alone. And it's already known. I already know that the biology is funny there. But the Himalayas, it's really the remnants of the Tethys Sea. So at any rate, the bottom line is that he found fossils like this one's called Pakicetus. The Paca comes from Pakistan. And there's Ambulocetus and Rhodocetus and whatever. Anyway, okay, that's a big, stupid digression, but phylogeny, it's a way of looking at evolutionary relatedness. But I think, it's also interesting to imagine conceptual phylogenies. And if you look at a book like the Alberts book, the way that it organizes biological knowledge is oriented towards, for example, a chapter on DNA. It's got a chapter on protein structure. There's a chapter on motility, this, that, and the other thing. And I'm just saying you could imagine an organizational scheme in which you say, well, I'm going to consider all the molecules which are allosteric, and that'll be my unifying organizational thread. And I don't care if that causes me to talk about hemoglobin and an ion channel in the same breath, because they're both molecules that exist in two states that are tuned by ligands. And that's the way we'll think of it. So what I'm going to do is I'm going to talk you through some examples of the allosteric concept, and I'll also do a couple of calculations. So I want to start with ligand-gated ion channels, and in particular, the case study of the nicotinic acetylcholine receptor. So this is a pentameric ion channel. Here you see the sort of schematic of this, and this is the drawing from David Goodsell, where what he's done is he's taken the PDB structure and the way he works. I've worked with him on 2 books now. I interact with him all the time. He basically takes atomic models and then he does watercolors. And he's an amazingly talented guy. He's a biochemist. He's a professor at Scripps, in case you don't know that. And he got interested in the fact that people were doing proteomics and had the numbers to be able to do pictures like the ones that Ted showed us. And so it was really a scientific endeavor that made him undertake this. So at any rate, he has a thing called the molecule of the month, and it turns out that nicotinic acetylcholine is one of them. You might have noticed all the molecules that I showed before, every last one of them was a good cell drawing. Each one of them has been a molecule of the month, okay? And they're all allosteric guys. You can see that there's 2 binding sites for acetylcholine. And what I want you to note is that there's a closed structure and an open structure for this channel. And so what I want to do is I want to think about the statistical mechanics of these kinds of curves. So this says normalized current, and on this axis it says acetylcholine. And so the concept is that you have a membrane, and in that membrane you have these channels. You take an electrode and you attach it to a little patch of membrane. You apply a little bit of suction pressure. You have an electrode in the pipette, and you have a second electrode going into the solution, and then what you do is you measure the current that passes through the channel. And you do this as a function of how much acetylcholine is present. So what do we see? We see that when there's no acetylcholine present, there's no current. This is current on this axis. When there's a lot of acetylcholine present, the channel's open. And in between, you have sort of open some of the time. Okay, and what this looks like in terms of the kinetics, I already drew yesterday. The kinetics is like this. It's just going up and down. It's open, closed, open, closed. So that's what a trace looks like of an ion channel. It's going back and forth between the open and the closed state. Now, why are there 5 distinct curves? There are 5 distinct curves. This one says wild type. And then they had a family of mutants that correspond to basically making specific amino acid changes to one of the subunits. So you can see that this thing is interesting. It's got one unique and then a couple of pairs. They make some amino acid changes, and then they measure the current. I want you also to just note, because it's going to come back as sort of a haunting feature of the data, is that they plotted normalized current, okay? And I'm very bothered by that because it does not actually reflect on the absolute open probability, unfortunately. And I'm going to show you what I realized as a result of that. So let me put up the board and just do a little calculation in the spirit of yesterday, just to show you how we could use exactly the same statistical mechanical ideas to try and reason out the behavior of an ion channel. Okay, so I'm going to do the same thing as yesterday. I'm going to do states, and here's my ion channel. And for the moment, I'm going to do the easier case of only one binding site. And here are the 4 allowed states of my little toy model. There's 2 closed states, closed and closed but occupied by ligand. And then I have open and occupied by ligand. So then I'm not going to write down all the multiplicities and all that stuff. I'm just going to straight away write the weights. So first of all, the weight of this state is e to the minus beta, and then the energy of being in the open, oops, closed. That should be a C. Energy of being in the closed state. And then here, I'm going to write e to the minus beta epsilon closed, and I'm going to have C over KD closed. Now, if somebody wants to talk about this further, go look at your notes from yesterday. You will note that we ended up with, for example, P over NNS, where P was the number of polymerases and NNS was the number of positions on the genome. Now I'm going from one dimension, where NNS is the number of sites on the DNA, to 3 dimensions. But when all the dust settles, this is what you get. Okay, here I have e to the minus beta epsilon open. And finally, I have e to the minus beta epsilon open, and then C over KD open. So what's P open? Does anybody want to tell me, based on what I've written, what's the probability of being open? Any thoughts? So I should use these two states in the numerator, right? So I have e to the minus beta epsilon open, 1 plus c over kd open, and then over e to the minus beta epsilon open, 1 plus c over kd open, plus e to the minus beta epsilon closed, 1 plus c over kd closed. So that's the result. And let me also just say that if I feel like it, I hope you don't mind, I might choose to write this like this. For the moment, leaving it kind of obscure as to why I would do that. So this is the open probability as a function of concentration. I want you to notice something. What if KD open and KD closed are equal? What happens? It cancels out from top and bottom, and P open does not depend on concentration. It's very interesting and very deep, in my opinion. So a key feature of the MWC model is that the binding of the ligand will have a different KD in the two states. If it doesn't, the thing doesn't work. Okay? They're different. No. If they're not, even if they're not the same, notice what happens. Because this factor, and that factor will be common. So I'll get a P open that's non-zero, but constant. Right? It will not depend on C. And indeed, there's something very interesting here that is related to my criticism of my buddy Henry Lester, who plotted here normalized current, because he tricked us. Because I'm going to claim straight away, it ain't true that the thing goes to 0 open probability at 0 ligand, nor is it true that it goes to probability 1. I know. But I went and told him something, which I'm going to show you in a few minutes. He's like, how do you know that? I said, I know it because. All right, so the bottom line is that what I want you to take away from this is that we can write down a statistical mechanical description of this relatively straightforwardly, and it gives us an expression for the open probability. Note also that it is not a Hill function. So I don't want to be dissing too much on Hill functions, but you know, Hill functions are a kind of binding function that you can only really use if it merits it. In other words, the binding properties have to actually deserve that. Absolutely, I'd love that.
So, you know, for years, there was these two camps on how one should view allostery. It was Koshland's way of looking at it and Monod's.
Yes.
And I'm wondering, is this sort of at the heart of a difference, or do they converge on exactly the same?
Yeah, okay. So Ted's asking about another one of these friendship-ending battles that happened. There were two classes of models. One of them was Monod, Wyman, and Changeux. Another one was Koshland, N, and F. I can't remember who they are. Bear with me for a sec. Sorry. But I'm going to just show you something. So my claim, Ted, is that I think it's relatively straightforward to see the connection between the two of them. And in fact, Manfred Eigen figured this out in the 60s. He realized that they're all basically the same idea, and here's the deal. Oh boy. In a nutshell. So I don't know, this is maybe getting a little ahead of ourselves, but hopefully I can. Okay, this is kind of a private discussion with Ted, just for a moment. So let's imagine that you have a dimeric thing with two binding sites. Monod, Wyman, and Changeux say that it's all or nothing. Either you're both in the inactive state or you're both in the active state. And so here there's 8 possible states. And I'm going to use this right now for the ion channel, because note it has 2 binding sites. So we're going to have 8 states to think about. K and F had a different idea. They said whenever you bind the ligand, you'll actually transition that particular guy. That was their concept. And this is Eigen's generalization. which you can include all of those possible states, you can see this is going to have more parameters than this and that. For me, I don't feel very invested in all that personal battle. But Changeux still, feels passionately about MWC. For me, they're all the same thing, which is you have two states and the ligand tunes them. But then it's particulars. It's kind of like, Heisenberg 2D versus Ising versus blah, Potts model. I don't feel that passionate about it. Okay, so moving on. So I just showed you how to do the case where there's one binding site, but here's the 8 states for the case where you have 2 binding sites. All right? So closed and empty. Closed and bound on the left, closed and bound on the right, closed and doubly bound. Open. Open bound on the left, open bound on the right, open doubly bound. What I want to do is I want to just comment on one of my favorite topics, which came up already, but I can't remember who brought it up. Data collapse. Yeah. So to me, this is like one of the, I still remember when I first learned about this, and it really blew my mind. So I'm going to show you 3 examples from physics and chemistry that you might have heard of before. So what this is a classic paper by Guggenheim, where what's being plotted on this axis is the normalized temperature, and this is the normalized density. And the reason I like it is that I find it very compelling and also bizarre that neon and argon and krypton and xenon and nitrogen and oxygen and CO and methane, I think, that their data will all fall on one universal curve if you find the natural variables of the problem. In other words, if you go to a data book and you plot the equation of state of each of these materials, they'll be different. But if you choose the right scaling variable, they'll all fall on the same curve. The same thing happens for magnetism. So this is from Gene Stanley. There's a bunch of different magnets here, different materials. And this is the scaled magnetization. That's the scaled temperature. And then these are a bunch of different superconductors. All the data falls on a master curve. Again, what's my interpretation of that? My interpretation is that means you found the natural variables of the problem. So let's check out the ion channel data. So here's the data I showed you, okay, the data points. The curves are using that equation, and this is all that data when I use a new variable, which I'm going to call the Bohr parameter. And this is Bohr not as in Niels Bohr, but as in Christian Bohr, his father. So the Bohrs have been kind of prolific, ladies and gentlemen. So Christian was hot stuff, okay, Niels Bohr's father. Then he had these 2 sons. Niels is the one most of you know about, but his brother was a super famous mathematician and also on the Olympic soccer team. Then Niels Bohr has some kids, and his son wins a Nobel Prize, and then, like your buddies, somebody's buddies that works with Henrik, your buddy, Henrik Flueveberg, or however you say his name. There's all these Bohrs that, I don't know, where are you? Yeah, you know these guys. They don't ever stop. They just keep, they're like the Bernoullis or something. Anyway, so I'm going to call it the Bohr parameter for a reason you're going to see in a few minutes. But here's the thing. The idea here is that if you want to figure out where to put a given data point, my claim is that what you should do is calculate this parameter, okay? So what I do is I take a data point. I know where the concentration is because it's this point, so I plug that in right here. Then I have this factor of 2, and then the epsilon is this epsilon. And that tells me the x-axis value I'm going to take for a given data point. And that's it. So the blue points correspond to this guy. The purple points correspond to that. The orange correspond to that. And so on and so forth. And what is my point, really? My point is that the data collapses onto a master curve. And I'm going to basically repeat the same motif again and again. So I'm going to show you like 4 examples, then I'm going to stop. Okay, so next example that I want to show you has to do with hemoglobin. So I don't want to go through the details of writing down all the states, but the bottom line is that one of the main competing models for hemoglobin is as an allosteric protein that exists. Probably most of you have heard this. It exists in the so-called T state, which is tense, and in the R state, which is relaxed. And depending on whether you've got much oxygen around, you'll favor one or the other. And I can never remember which is which, to be perfectly honest. So here I'm showing you a dimeric version of hemoglobin, which is called dimeglobin, and you can see how the math would play out. And this is the equation that results for the actual hemoglobin. So what it's saying is number of oxygens bound as a function of oxygen concentration is given by this equation. And you can see, once again, the structure is the same as the MWC structure that I just wrote down. So if you work through this, okay, so a little bit of a digression now about Bohr. So what Bohr was interested in was how hemoglobin worked. And he made this discovery, which is now known as the Bohr effect. And the Bohr effect has to do with how, as you change the pH, the binding curves for oxygen will change. And this is true for many other chemicals. In other words, it's not just the pH, but you can also put in BPG, which is a kind of famous one. You can put in carbon monoxide. There's all these other things, and they will change the binding curves. So you once again get families of curves. So here I'm showing you probably, I've tried to ask around probably the most reputable recent experiment. And what's shown here is wild type hemoglobin here for different pHs, okay? And this is basically the number bound, but in a little bit of weird units. And this is the partial pressure of oxygen. Here, this is binding of oxygen, but in the presence of an effector. This is in the presence of a second effector, and then these guys are with two effectors at once. So there's this huge quantity of data, all right? And if what I'm telling you is correct, it ought to be true that all this stuff really should be smushable into, a couple of master curves. But I first want to tell you about the sad part of that paper, at least in my opinion, okay? What this is a table from that paper in which they took each one of their data sets, used this model, and fit the KDs. So these are the two KDs. And then epsilon, my L, or their L is my e to the minus beta epsilon, and so on. And what I find shocking about it is they fit a different set of parameters for every single different experiment. So like for me, I don't know, I'm not trying to insult them. I think their experiments are amazing, but it's very muddled because it doesn't accomplish anything. And here, now coming back to what I would say is if you have an effector present, you actually need to account for that in the statistical mechanics. So notice, this is a different equation. I have C and X because there's two species present. These guys, they fit it to just the oxygen. So it's a bit like just grabbing a Hill function off the shelf and fitting some arbitrary sigmoidal curve. It's just a curve fitting thing. And they also did fit Hill functions. So what I wanted to say is that using one minimalistic set of parameters, you can fit all the data at once, basically. All right? So just trying to encourage you to see kind of the unity of this. Okay, now I want to turn to another example. So I don't mean to be fire hosing you with 10 different examples, but so far what I've told you is an ion channel. I think it's fairly straightforward. Ion channels sit in the membrane, they open and they close, and how probable they are to be open depends on the presence of a ligand in the case that I told you about. The second example, enjoy your hemoglobin. Okay, so you saturate the hemoglobin, it goes off in your blood, and when it gets to lower partial pressure, it dumps the oxygen. So that was my second example. Now I want to tell you about bacterial chemotaxis. And this is really the case study that caught my attention and motivated me for this. And what I'm getting ready to tell you, I already told Sid, is I am a confessed Ned Wingreen groupie. Okay? So Ned Wingreen wrote some amazing papers on this stuff, and that's what I'm going to tell you about now. Two things from him, and then I'll kind of try and summarize. So what's bacterial chemotaxis about? I don't want to belabor it. I'm sure all of you have heard about it before, but the rough idea is that E. coli, for example, will march along for about 30 microns. They'll put out their flagella. They'll go to a new orientation. They'll go off for another 30 microns. Another second, roughly, per run. All right? And the observation, which we already heard about, is that depending on whether or not conditions are getting better, how often they flop will change. In other words, if conditions are getting better, then they'll be less likely to flop and go off in a new direction. So Howard Berg likes to say that E. coli is an optimist. So, you know, if things are getting better, keep going. If things are getting worse, well, try a new direction. All right? So the concept of that is illustrated here. So this is receptor activity as a function of chemoattractant. So if you don't have much chemoattractant around, then the receptor is active, which means the signaling molecule goes to the motor and says reorient, and you reorient. If there are chemoattractants around, you suppress reorientation. All right? So that's the idea. It's implemented by these kinds of, this machinery. It's, you know, you could spend 2 weeks in a course talking about this, and I'm not going to. But for our purposes, what you really need to know is there's a receptor, it binds ligands, and when it does or doesn't bind ligands, it'll actually, it will stimulate the phosphorylation of a messenger molecule that goes off somewhere else and tells the motors either to turn or not. Okay, so that's the rough idea. And once again, Howard Berg and Victor Sorek, they did experiments on a bunch of different mutants. They have a whole series of different mutants and a whole series of different ambient concentrations. Both are interesting. So let me just comment on that. So if you look up in the sky and you look to the North Pole, or look to the Big Dipper, and you follow it over to Polaris, the photons arriving at your eyes when you see the North Star are separated by 3 kilometers on average. Okay? You should check me on this. So you're looking at a star, and each photon, the light is so dim that the photons are 3 kilometers apart. Now, that doesn't mean they're very far apart in time, right? Because light goes fast. But they're 3 kilometers apart. If you go out on a glacier in the middle of the day, you know, in the French Alps in the height of summer, if you don't wear glacier glasses, you know, you'll end up snowblind because there's so many photons. And the reason I'm telling you this is that our eyes can adapt over a very wide array of light levels. And the reason that's important for chemotaxis is because an E. coli cell can detect a 0.02 micromolar per micron gradient, okay? But over a 4 order of magnitude absolute concentration range. That's impressive. In other words, so most things you'd think up that are based on binding, you have a binding curve, you hit the KD, and that's the sweet spot. So how do you construct something where you have a movable sweet spot is really kind of the question. And so what these experiments attacked was the question of what happens at different ambient concentrations, and then what happens when you turn off the machinery for adaptation. So you can see there's families of activity curves. I'm not going to go into the details, but if you want to learn about it, go read the papers by Berg and Sorek. They're amazing. So along comes Robert Endres and Ned Wingreen, and there's another guy, I forgot his name, with a sort of, I think it's a Spanish name. Who is it? Juan what? Yeah, but what's his last name? That's right, exactly. Okay, so what they realized is that the activity of these receptors can be written, think of them as MWC molecules. The receptor is either active or inactive. And it depends on the presence of the chemoattractant. Same stuff all over again. All right, I'm not going to bore you with it, literally, bore or otherwise. But if you go look at their paper in PNAS, shown right here, this is the curve. This is the one that really caught my eye, that got me going on becoming a groupie, okay? Because as I told you, I always thought this idea of data collapse in the stat-mech critical phenomena setting was really something. And it's also why I like Hamilton-Jacobi theory in mechanics. It's like tricks for finding the right variables to me, or normal modes. I mean, what's better than normal modes? Let's just think about this. 2 masses. Let's see, I can draw it so that we'll all be on the same page. So I have mass, spring, wall. So, you know, if I pull just one of these masses, the behavior is weird, right? It's gross. But I do know that if I do this, I'll get that. And also if I move them both like this, then I'll get that. So there's two special variables, the symmetric mode and the antisymmetric mode. So that's the natural coordinates for that problem. And so here, what Ned and those guys figured out is that the natural coordinates for thinking about this is this thing that I'm calling the Bohr parameter. The reason I call it the Bohr parameter is because in hemoglobin, the data collapse is the Bohr effect. In other words, it's taking the Bohr effect and bringing it all onto a master curve. Okay, so groupie hood part 2. So this is another paper that came from Bonnie Bassler and Ned Wingreen. I'm not going to go into details again, but if you don't know about their stuff on quorum sensing, well, then you should go learn about it. The story, the most interesting for me biological story has to do with a bobtail squid in Hawaii. So these are these little tiny squid, and they basically take on Vibrio, I forgot which one, maybe fish or eye? The squid, though? I think you might be thinking about Bonnie. Hers, yeah, okay, no, but anyway, I don't know. I think Bonnie works on Harvey eye, but maybe she works on fish or eye, I don't know. But at any rate, and it's an amazing microbiology story because they only take on the right bugs. They reseed themselves every day. And when at nighttime, here's the just so story part, by the way. So Art, listen well, please, and tell me like if you agree with me that it would be cool to dig into the biology better. So the claim that's made is that these squid have these microorganisms that light up. You can see they light up and glow because, here's the part, the because is the just so part. Because fish that are deeper in the water at night looking up will actually see a shadow if the squid don't have their lights on. It's plausible. I mean, I know for a fact that there are all sorts of organisms that do dynamical camouflage. So if you haven't, take down a name. Go on YouTube. I'm giving you a name right now. Roger Hanlon, Hanlon. Go look him up after my talk today. You will see the most amazing video you've ever seen of a cuttlefish changing its colors in real time. But back to the squid thing, I don't know. Like, I just haven't been able to dig into the actual literature that demonstrates, like, how do you do that experiment? It's not that obvious, but anyway, okay. So these bacteria, when they get tightly together, they decide to turn on light, all right? And there's receptors. They respond to chemical stimuli. That leads to a cascade of gene expression. And the thing that Bassler and company did is they examined all sorts of mutants. Here's the list of mutants. And they looked at the activity as a function of one of the signaling molecules. You know where I'm heading with this, don't you? So we have both mutants and different chemicals. And so in their paper, I can even read it. It says, we had reasoned that the dark phenotypes of our LuxN mutants could stem from, 1, a defect in the ability to bind AL1. Okay, let's just check out what the words are. There's a receptor, it binds some chemical, and they're saying, well, one of the ways you could be a mutant is that you don't bind in the same way. That makes sense. 2, a bias favoring the kinase state. That's damn interesting, because it's not talking about binding anymore, it's talking about the state of activity. Remember, I was telling you, these receptors, they're either active or inactive. So that, for me, is the parameter epsilon that controls, whether you're highly active or not. C, a defect in signaling, or D, some combination of both. Okay, here's the part I like. The method of data collapse provides us with a powerful tool to distinguish among these possibilities. So in their paper, in the appendix, unfortunately, they wrote down the wrong equation, which Ned is very embarrassed about, but here's the right equation. And it's the same equation I wrote a few minutes ago for hemoglobin. It's the Bohr parameter. Note, there's the data collapse. Very interesting. Here's a challenge for bioinformaticists. I carry around, so okay, the challenge is, do you think just by goofing around with data fitting and data mining and high throughputing and whatever else it's called, massively paralleling, that you could figure out that this is the right variable for the problem? I have my doubts. Like I've been offering to buy somebody dinner in Paris if they can prove to me that by nothing more than just somehow goofing around with all the data, they can figure out that the natural variable of the problem is the Bohr parameter.
Would that include flying them to Paris?
Maybe, but perhaps not business class. But a good restaurant.
What's spending a week then on it?
Yeah, well, yeah, so where are you coming from, Toronto? Okay, yeah, I'd probably be game for that. So I was going to say, I also carry around, if anybody wants to see it, it's kind of fun to see. I carry around Tycho Brahe's data for the position of Mars, okay? And I want you to think about what Mars does. Mars goes forward, and then it goes backwards, and it goes forward again. But what's Tycho Brahe's data? This is some, this is a guy without a telescope on an island in cloudy Denmark. And he had these giant protractors, basically. And his data is this angle, that angle, date, and time. That's his data. And once again, it's like I want to buy somebody dinner for that too. I know biology is complicated, but I want to see somebody take that data and figure out Kepler's laws of planetary motion. I don't know. It's not so obvious. Okay, so last example. So I want to come back to what we talked about yesterday, which was transcription. So here what I'm showing you is the states we looked at yesterday, but now I'm acknowledging the fact that my repressor can actually be in two states, inactive and active. And I'm saying we can do the statistical mechanics in that case. And here what I'm showing you is that there's an MWC description of the relative stability of active and inactive states. And the point of this was, which I had this part hidden at the beginning, but here's the data, a bunch of different mutants, and here's the data collapse that you get by, again, just simply using the Bohr parameter. So, I kind of kiddingly say right here, I should have said it, for physics types, I'd love to hear from people in biology, but for me, I love mutants. I really do. I think mutants are one of the most interesting things in biology. But I hate tables like this because they don't mean anything. Like I didn't do the work, so I don't know what all the letters mean. Oh, I guess I kind of do. It's like they have A and then some number and then some other letter. So it's, it's a particular amino acid substitution. But I kiddingly think that we should reorganize our -80 freezers with our Eppendorf tubes just labeled with, epsilon in KT units. So, this is the -14 KT mutant, and that's the -12 KT mutant. Because, from the standpoint of MWC, I don't care what amino acid it was. It's just some single change in the binding energies. Yeah. Okay, so last thing, I'm going to be done so early. That's good. Is I want to come back to nucleosomes. So far I've told you about allostery from the protein point of view, and now I want to focus a little bit on DNA and its conformations. So I already, sometime, it feels like we've been in this room for a week, but I guess we have. Yeah, that's why. So here's a nucleosome. The blue part is the histone octamer. The orange stuff is the DNA. I told you that people have looked in flies and humans and cancer cells and Drosophila and worms, the list goes on, in yeast, at every single gene, and they've asked the question, where are the nucleosomes? This is two representations of the same data. This is gene number as you go up this. There's 7,000 or so genes, or 6,000. This is all the genes in yeast. And what you're supposed to, and green versus red tells you about where nucleosomes are and where they aren't. Nucleosome-free regions and nucleosome-occupied regions. And so what I wanted to say a few words about is the question of accessibility. So we can think about a single nucleosome. There's a beautiful experiment that was done by Pollock and Whittam. Well, what they did is they asked the question, I'll use my sweatshirt. You imagine that there's some binding site on the DNA. And you're going to ask the question, how easily do I get to a binding site as a function of how deeply the site is buried within the DNA? In other words, this site right here is a heck of a lot easier to get to than this site, which is easier to get to than this site, and so on. So the DNA is wrapped 1 3/4 times around. So once again, you can imagine an MWC type mentality, because here I have the closed state, here I have the open state, closed, empty, closed, occupied, open, empty, open, occupied. Just the same thing I've been doing over and over again. I'm not going to keep boring you with it. And what I wanted to say is, and here's the outcome of the measurements that were done by Pollock and Whittam. So what you see here is this is called the equilibrium constant for conformation. And this tells us about how easily we can get to sites that are buried at different depths within the nucleosome. And I want you to note that it's an exponentially decaying function of the burial depth. In other words, the more deeply you get, each time you go, something like 20 base pairs, you get 1 or 2 orders of magnitude change in the equilibrium constant. So you can use this MWC stuff to calculate it. And here what I'm showing you is the probability of binding, or the probability of being active, for 3 different burial depths. And I'm once again just showing you, this is not experimental. The other ones were experimental data. But this is just showing you that this all collapses onto one master curve. And the reason I got amused by this is that I'm not only a groupie of Ned Wingreen, but I'm also a groupie of Leonid Mirny, who's a guy at MIT. Super sharp guy. So he wrote a beautiful paper, as did Alexei Igoshin. I think that's his name. Do you guys know him? He's at Rice. Super interesting guy also. These 2 papers right here are really great. And the idea of both of them, it's a hypothesis, okay? But the hypothesis is that you have a closed state of chromatin and an open state of chromatin, and that the binding of ligands has a different KD when you're in the closed and the open state. So, this is the ion channel case all over again. I'm not going to, you can see there's 8 states, because it's empty, bound left, bound right, doubly bound, closed, open, bound, not bound, bound left, bound right, doubly. And the reason that that's interesting, I think, is that it leads to a hypothesis about post-translational modifications of histones. So the suspicion is, so first of all, this is the probability of being active. And what I would say, that I think is what Mirny was suggesting in the first place, is that stuff like methylation and acetylation shifts the parameter epsilon. But that's really the way that one could think of it. So once again, rather than getting all hung up on some detailed chemistry ideas, one might be able to say, well, this is an MWC type picture. And really what acetylation is like changing the pH in the context of hemoglobin binding to oxygen. So I like that idea a lot. Leonid tried to make it explicit. Here you see, this is the modified nucleosome binding curve. This is the unmodified binding nucleosome as a function of the protein concentration. So I think this is very provocative. I've tried hard to find the data, and nobody's done the right experiment yet. Right with quotes on it, meaning right something that I want. Okay? So it's not that obvious how to do the experiment. But what you'd like to do is measure the binding curve systematically as a function, for example, of the state of acetylation, that kind of thing. Okay, so I think I'm basically done. Let me read this part, because I think I was trying to say something useful. So fascinating, yes, and unexpected that hemoglobin and the work of Niels Bohr's father comes full circle to suggest a line of inquiry on chromatin and gene regulation. And then this really drives home my point that physical proximity of topics sometimes reveals biological insights that are completely invisible when viewed from the perspective of biological proximity. Said differently, hemoglobin workers and chromatin workers don't talk to each other, really. Not much. I mean, sure, there are exceptions. I'm not trying to make some grandiose generalization, but who would have thought that the obscure Bohr effect would serve as a model for chromatin modification? Well, okay, I alluded to this yesterday, actually, and so I want to just give you the quote, the way Feynman said it in his Nobel lecture, because I think it's a really smart thing that he said. Theories of the known which are described by different physical ideas may be equivalent in all their predictions and are hence scientifically indistinguishable. So think, Schrödinger's wave equation for the harmonic oscillator, matrix mechanics for the harmonic oscillator, path integral, you get the same answer. Or Lagrange's equations versus free body diagrams, sum over all the vectors. Or Lagrange, get the Lagrangian. You get the same answer if you try to do a harmonic oscillator or the planetary motion or whatever. However, they are not psychologically identical when trying to move from that base into the unknown. So the argument I was trying to make yesterday is, theory, genetics, biochemistry, cell biology, molecular biology, developmental biology, evolution. Each one has its own psychological inequivalence, perhaps, when trying to go into some new problem. I've had a lot of fun in my life learning science. It's just been one of the great thrills of my life, truly. I'm not just saying that just to sound cool or whatever. It's like been one of the great pleasures of my life. And one of the things that I've loved, and I mentioned this earlier in the week, it just surprised me. I was so naive. I just didn't know anything. I wasn't expecting, when I learned about RLC circuits, that the harmonic oscillator was going to come back at me again. And I just thought that was so great. So, everything's a harmonic oscillator. And, of course, that's kind of true if you start in equilibrium and you perturb locally. Okay, there's exceptions. Why am I saying this? Because today, what I was trying to argue is, in a way, that everything is an MWC molecule. Yes, of course, this is an exaggeration. But allostery is a super dominant thing in biology, and there's a beautiful mathematical framework for thinking about it. And it's very different from Hill function-ology. So Henry Lester fitted, I don't know if I showed it to you, but he fitted the ion channel data with a Hill function. There's no way to get a data collapse with that. It doesn't even make sense, other than as a fit of data. I guess I also wanted to say, again, I apologize to those of you who've seen this before, but this is the classic picture of the Tacoma Narrows Bridge. This is Hurricane Sandy from a year ago. This is the Juan Fernandez Islands in Chile. And my main reason for bringing all those up is that, yes, all of that stuff is made of atoms and molecules. I accept that. And yet, I have absolutely no interest in the atoms or molecules. And one of the things I wanted to say yesterday was that kinetic theory is super cool, and I love it. But thermodynamics is, in some ways, even better, in my opinion. And what's a little odd about that is kinetic theory is the mechanistic thing. You have a box. You want to figure out how much pressure there is. Well, you say, oh, it's molecules. They collide. I compute the impulse. I add them all up. That's mechanistic. But PV equals nRT and, Maxwell relations and all that stuff, it transcends the details. I don't care what the details are. And so I feel that, in a certain sense, that biology needs more of the thermodynamics, elasticity, hydrodynamics, coarse grain, don't care which residue kisses which other residue thinking. And look, okay, so probably 99% of people don't agree with that, but how about letting 1% of people try to not worry about which residue kisses what residue? The MWC model has all sorts of cool and interesting properties, and I want to call your attention to one of them, which is the effective Hill coefficient. So these things have Hill coefficients. And interestingly, they will be weird things like non-integers. But that doesn't mean that there's 2.8 oxygens that bind hemoglobin. In my opinion, it means you have the wrong model. Somehow thinking about a Hill coefficient is maybe just misguided. So anyway, if you want to do some homework, you can calculate all these things. It's got the dynamic range. It's got the, the dynamic range is in there. The slope is in there. Where the half point is. There's all these features, and they're coupled. And that's something quite fascinating. I should have shown that to you. This is something Mitch Lewis appreciated here. So notice this guy has very little leakiness right? When there's no inducer around, there's no expression. But it's also got low dynamic range. This guy is leaky, but it's got quite a lot of dynamic range. And what I'm telling you is they're not independent. You can't have it both ways. It's super interesting. So, and that's what these little equations were showing. In other words, if you go look at these equations in detail, you can see they're linked. You cannot get zero leak and high dynamic range together. Okay, I'm done. I don't know what we're going to do for a half hour, but we can do stuff. But I wanted to just finish with a few things. So one is, so I still am worried I'm not going to say your name right. But there's a very famous course that's run every summer. There's many famous courses, such as the developmental biology course. It's called embryology. There's the physiology course. There's the microbial diversity course. There's the neurobiology course. These are 7-week courses that are given in Woods Hole, which is in Massachusetts. And so I wanted to make a plug for all of the courses. The application deadline's like in a month, I think, and for the physiology course in particular. So it's a 7-week course. The first week is a boot camp. It's about half physicists and half biologists as far as the student body goes. In the boot camp, there's 2 days of mat lab, but very intense. 2 days of biochemistry where you basically take squid and you purify the protein kinesin, for example, and then 2 days of microscopy. There's $5 million worth of loaner microscopes there every summer. You know, best turf, best confocal, best everything. Do you agree? Okay. So in the hallways, there's just all these huge crates that are there from Zeiss and Nikon. And then there's 3 2-week research rotations. So during each rotation, you have 3 or 4 faculty members. Each of the faculty members takes roughly 9 students. So next year, there will be Jennifer Lippincott-Schwartz. Jitu has been a faculty member the last few years, Jitu Meyer. Wallace Marshall, Jennifer Lippincott-Schwartz, Joe Howard, Dan Fletcher, Adam Cohen, Jody Nunari, Nicole King, I forgot, Bob Goldstein. Yannick Kondev. There's a bunch of fun, interesting people. Every day there's a speaker. The list of speakers is pretty amazing. So I personally would encourage you to think about applying for these courses. And I don't know, I mean, it's not, I'm not going to get anything out of it. I'm just telling you, in my opinion, this is the best scientific thing that I've done. Then I've talked about a lot of stuff all week. One of Ted's colleagues, I don't know how well you knew him, and how pretty well, so I don't know if he was a great guy or not, but I think he was an interesting guy from what I've been able to read. Anyway, he made a wonderful remark that has meant a lot to me. He says, we all know that the real reason universities have students is in order to educate the professors. And there's few people that that's more true of than me. And so even though I've talked about a lot of stuff this week and said I this and I that and all that, that's really crap. So there's so many people that have been critical to the things that I've told you. So Yannick Kondev is a guy I've worked with. We talked on the phone every morning for 10 years, do calculations every day. The noise stuff that I showed you was Daniel Jones and Rob Brewster. I love that. I think they really did something very clever. The stuff about copy number was Franz and Brewster. Hernan Garcia was a huge part of all of these things. He and Lynn Hahn, they came to me as grad students when I moved to Caltech. I already told you, I don't even know if I'd seen a pipette. Like literally, it was that bad. And I started a lab. So, it's lunatic move. And they were lunatics too because they joined me. So, we started from nothing, literally nothing. When she came into my lab, there was nothing. I have a picture of her with nothing. And she did very cool stuff. So at any rate, it's been great working with those people. And also, as I mentioned, I'm a Ned Wingreen groupie and a Leonid Mirny groupie. These 2 guys really have inspired me with their MWC stuff. And Jean-Pierre Changeux has really been very helpful. Julie Theriault, Yannick, Hernan, Nigel Orme, been an amazing group of collaborators. I even get to work with superheroes. So this is Ron Milo. He's the guy who has the BioNumbers website and with whom I'm writing a book on cell biology by the numbers. And this deeply saddens me to show you. It's a picture that was taken in China. That's John Whittam, who unfortunately died a few years ago at the young age of 54, but was very critical to stuff I've done. And then this is Sarah Marzin, with whom I did the MWC stuff. So with that, I don't know, we can chat for 25 minutes, or we can maybe have a general discussion, or we can stop, whatever.
idea, but you only if you're up to it.
I'm up for most anything.
Because I'm interested to listen to.
Yeah.
Could you tell us in next 20, 25 minutes what you're working on now?
Sure. You want me to do it like even using PowerPoint-ish stuff?
As you wish, as you wish.
Well, are you sure it's okay with everyone else? I mean, I can always close my eyes and like this. You can decide. I don't know. I don't know. We should let people escape if they want to. Okay, yeah, I think I can quickly do a few things. So, does anyone have any questions? I mean, while I'm looking?
So one issue is, so in order to get this collapse plot, one is to measure especially things either in equilibrium or without delays.
It's a very good point.
So can you extend this, I mean, to systems which are, I mean, for example, in CHEMOTAXIS problem, I think the change in the concentration will affect the change in motion only after some time.
Yeah.
So what exactly were you measuring this?
Yeah, so this is a great point, and it kind of is related to the title of yesterday's talk, which you weren't here for, I don't think, but Napoleon is in equilibrium. So so the question really was, hey, you know, you're kind of using equilibrium ideas, and clearly these things are dynamical. And so there's a whole parallel universe of things that people have done for the dynamics of MWC molecules. All the measurements that I'm telling you about, I think it's legit to think of them as being equilibrium measurements. So I think that's fine, but you're absolutely right. You know, there's dynamical things where you care, and so in that case, instead of writing down equilibria, you do, again, what I was doing yesterday, you write down rate equations, there's constants, you can figure out things like the transient times and that kind of stuff. Yeah, so anyway. Okay, I'll just quickly tell you one of the things that I've had a lot of fun with, which is mechanosensation. So we've been thinking about this in bacteria. So, you know, it's clear mechanosensation is everywhere. And the quick idea for my purposes is probably best illustrated by thinking about something which is slightly rude, but we each have 2 kilograms of bacteria in our guts, you know? And so if you wake up in the morning and you use the facilities, then what you're doing is you're basically taking bacteria which are in a salty solution and you're putting them in fresh water. So what will happen? If you put bacteria in fresh water, and I highly recommend if you have a microscope, next time you have a cut, you know, take your blood and put it in some PBS and put it under the microscope and then put some salt in. And what you'll see, have any of you seen this? The red blood cells will actually poke out little tiny protrusions. And this has to do with the osmotic effect. In the case of bacteria, what will happen is water rushes in. And the way to think of that is that the concentration inside of the cell is much more concentrated than outside. There's an osmotic pressure difference. Water rushes in to try and dilute out the high concentration inside of the cell. So what happens is that they swell. And in the absence of certain mechanosensitive channels, the cells will rupture and die. So I can show you a movie of this. So here you see the case of actually putting it into highly salty solution. Here what happened is the water from the cell rushed out, and it's shriveled up like a raisin. The next case, this is the one at around 5 seconds, you'll see the cell inflate. Okay, water was rushing in, and then at a given moment, the thing actually inflated. And then the last example is just to show you what happens in the absence of channels. So they pop. So we've been interested in the properties of these channels. How do they work? How do you actually mechanically gate an ion channel? So, so let me just say a few words about these channels. So I already told you about this one a little earlier. But we know a lot about the structures. So Doug Rees, for example, and Ian Booth have solved the structures. here's the mechanosensitive channel of large conductance, side view, top view in the closed state. And then this is the mechanosensitive channel of small conductance, side view. Yeah? No, definitely not. No. In fact, what we found, that's part of what I'm going to show you. What we found, in fact, is that most of the time they don't rupture. They die by other things like blebbing and by letting the DNA out and all sorts of things, longer term, not immediate rupture. There's tons of stochasticity, and I can't say that I deeply understand that. So, so the way you do measurements on these is exactly what I said a while ago. Here's a schematic of what I was telling you about. You take a pipette, you grab a little piece of membrane, here's the electrodes, you basically apply a suction pressure inside of the pipette, and then you measure the current, and here's what the current traces look like. Up and down, up and down, up and down, up and down. So that's what the current traces look like. And what comes out of that is that kind of stuff. So I showed you this on the first day. This was one of the experiments that really blew my mind and made me think about changing fields. I apologize. This all should be shifted down. But at any rate, what's plotted here? On this axis is the pressure in the pipette. And on the vertical axis is the probability of being open. And the thing that I thought was so provocative is that you have different gating curves for different lipid bilayer thicknesses. The C16 refers to the number of carbons in the tail of the lipid. So that was kind of the hint that got us started in this subject. And I'll just quickly tell you, this is kind of, for me, a very fun theory project that was thought up more or less exclusively by Paul Wiggins. And the idea is really interesting. And it's, to me, a good example of how fun it is to have different worldviews coming to attack given problems. Because a lot of people, actually, I think I've got a picture that will illustrate this. A lot of people think that the ion channels are opened up by trap doors attached to pulleys and strings and stuff. See that? Trap door, it gets pulled on. Like, you know, somehow there's actually literally a mechanical thingy that tugs. And what I wanted to say, what I thought was fun that Paul figured out, is that there's a much simpler way of thinking about what causes gating. And I can explain it to you super simply. So you have an ion channel, and you should think of the consequence of an ion channel as being a bit like what would happen if you put a bowling ball on a mattress. So if you put a bowling ball on a mattress, what happens? You get a dent, and the dent has a wavelength. It has a length scale over which the bed is deformed. So if you put an ion channel or any protein in a membrane, you have a perturbation with some length scale. So I want you to think of that. Now I'm going to act it out. I want you to think of that as an annulus of deformed material. Okay? So out here, that's the equilibrium thickness of the lipid bilayer. My arms are sloped, and everywhere, now I'm tracing out, I hope you can see, I'm tracing out a donut of material that's deformed. Now what happens when the radius gets bigger? I can't really do it, but if I had stretchy arms, then I would do this, and now what I'd have is a larger radius region of deformed material, but with the same length scale. So what's that mean? That means that I actually will pay an energy, a free energy of deformation to open the channel. And so if you like, you can think of the gating as a competition between two effects. One is the cost of the deformed material, and the other is the advantage, which is that if you gate the channel, you lower some weights. Okay, I don't know if you're used to thinking of, you know, an optical trap or a tension apparatus. Really, you can always think of that as a loading device with fixed weights. In fact, I have a picture on my computer. It's so cool. There's a museum in Paris called Art and Métier. You should go there. They have the apparatus of Gay-Lussac. And he has a box. And on it, he has lines that go through pulleys that are attached to weights. And that's literally how he measured, you know, the PV stuff, was with pulleys and weights. And so this is a fantasy, but I'm asking you to imagine that when the channel gates, these weights drop a little. And so I can compute the energy gain. That's a better world when the weights drop. That's got a minus sign. The potential energy went down. But the free energy of the membrane went up. And so that's the competition. So you can do calculations. I'm not going to bore you, but the upshot is this. You get a competition. It's nucleation theory. This thing is positive, and it's the cost of the deformed material. Notice that it goes like 2 pi r. All right? And then this part is the weights falling, and it goes like pi r squared. So tau is basically the force per unit length pulling down when the thing relaxes. And so you can compute the critical tension, and that's when the open and the closed state are equal. So let's see. Well, I guess, you know that there's a great joke about the mathematician was giving a talk, and then somebody asked him if it's obvious, and he sat down and he thought for an hour, and then he got up and he said, yeah, it's obvious. So, also Laplace, I have in my house, I have Laplace's 8 volumes or whatever of his celestial mechanics. I don't know if you've heard about it. It's amazing all the stuff that Laplace did in there. And he went to Napoleon, and Napoleon said a few things to him. One of them was, he said, I noticed there's no mention of God, and so Laplace told him, I have no need of that hypothesis. But the other thing is that Napoleon told Laplace, he said when he had a spare week, he would look at the book, and there's a bunch of it can be shown that's in there that took the likes of Gauss and others, a year to figure out. Anyway, okay, so is it obvious? I was going to say it's obvious in the sense that it's tension area, just like pressure volume. But no, I think you got to work it out. I don't know. It probably is obvious, but it's not. I calculate it. So I'll tell you how you calculate it. You basically, here, what you do is you notice that when the thing gates, you change the radius here, right here, by some amount, delta r. And in this channel's case, you go from 2 nanometers to 3 nanometers radius, so it's a lot. But the membrane is not, when you change this, the area of the membrane, the total area doesn't change. So all that happens is that these guys go down, but you use the conservation of area to figure out how much this guy moved by. And it's not much, but it turns out that the change you get is the area of the channel, the delta R, or the area change of the channel. So I don't know. You should work it out and see if you think it's obvious. Okay. So I don't know. I really don't want to just ramble on forever. But, all right, I'll just quickly tell you about something completely different. So let's see if I can figure out where it might be. Because I think you might be amused by the virus thing. So that's what I'll quickly look at. All right, maybe this has it. I don't know. Okay. So this is probably what you were driving at, perhaps. But so we think a lot about bacterial viruses in my group, which is a really interesting topic. And so one of the things we've been interested in is kind of comes from a challenge that David Baltimore made to me that just has to do with how little we know about what viruses are out there and who they infect. So, there's this famous tree of life, and yet we know that more or less for all the organisms we know about, there's viruses that go along with them. And there's all sorts of questions about what's the range of infections. So just to make it personal, the influenza that we fear the most, I think, is the influenza that gets mixed up in pigs and birds and stuff, right? I was thinking of trying to grow some tobacco plants and then to have tobacco mosaic virus in my backyard. But then somebody was telling me that TMV actually will infect 500 different types of plants. So it's actually pretty promiscuous. So we don't know much about this, okay? And so what we were interested in is trying to figure out what tree goes along with this. So I already told you about this paper the other day. And in this paper, I mentioned a figure, which is that there are 10 to the 6th microbes per cubic centimeter in the ocean. And there's something called the VBR, which is the virus to bacterium ratio. So if you take an electron microscope and you look at seawater, you'll find a bunch of bacteria, but you'll find a 10-fold excess in general of viruses relative to those bacteria. So, here in general, you can see that there's quite a few more viruses than there are prokaryotes. So we've been interested in trying to figure out, how does the phage tree go along with the so-called tree of life. Now, there's problems with this. How did this tree actually even get constructed? Well, back in the old days, it was based on morphology and anatomy and fossils and that sort of thing. And that's not always that obvious. You could make guesses, but it's not 100% clear who goes with whom. And a good example comes back to what I talked about earlier. Who are the relatives of whales? And there's still arguments about that, because DNA and fossils don't necessarily agree 100%, which is cool. It's interesting. But anyway, so this is mainly done using the ribosome, right? Looking at the sequence of the ribosomal RNA. The problem is that there's no equivalent of that for viruses. There's no universal marker that we know how to use. Also, it's very convoluted because of horizontal gene transfer, meaning that there's even mosaicism. So what we've done, and then I really am going to stop after a minute, but what we got interested in was looking at two natural environments. One of them is the termite gut, which may sound kind of obscure, but when we were in Periyar last week, it was really very interesting. We saw a bunch of dead trees, and then we saw mounds that were this high, termite mounds. Have any of you seen these before? So that was pretty nifty. So that's a termite, and it's a termite from the mountains outside of LA, and you can take a pair of tweezers, you can grab it right here, and you can pull out its gut, which is right there. And then if you squeeze the gut, it's full of all sorts of organisms at all scales. Protozoa, but down to spirochetes and bacteria and this and that. And it all has to do with managing wood and being able to eat wood. So we were interested in asking the question, what viruses are here? And the way we ask this question is using the exact same technique that I mentioned before, which is digital PCR. And the idea is that you basically divide the contents of the termite gut onto 765 chambers. And then you do a PCR reaction with two sets of primers. One is 16S to try and identify the microbe of interest. And the second one is the viral terminase primer that we were using. And then whenever you get a simultaneous hit, two colors, you know that you found a virus and a host together. Okay? And we just did this again and again. I should say Arbel Tadmore did this again and again and again. Recently, what we've been doing is the same thing in the context of the human mouth. So it's really very interesting. Our mouths are full of bacteria, hundreds of different species. And it would be very, from a standpoint of our diets and stuff, yours and mine are probably quite different. And mine is going to be pretty similar to my wife's in all likelihood, and probably even to the dogs that I live with. What? Yeah.
How things get started. They started the dog, they got him the baby, and they got on Bruce, and got on the mom.
Yeah. It's very interesting. Yeah, so, and unfortunately, we have a dog that sleeps under the covers every night, so it's a lost cause. But my question and so the bottom line is we have tons of viruses in our mouths that attack our bacteria. And kind of where I'm headed with this is, what are they doing? What are they up to? And maybe I'll just finish with a thought, which is that 150 years ago, Koch and Pasteur articulated this idea that microbes are pathogens. That was really, in a way, that was the end of cell theory in the sense that it was finished when Koch and Pasteur came onto the scene. Because until then, people didn't know that bacteria were cells in the classic sense of the word. But for 30 or 40 years, it was thought that microbes are pathogens. And it took the Dutch school primarily to figure out, what? Most microbes are not negative actors in natural environments. In fact, they're positive actors. So, for example, if you deprive your kid of all of their bacteria, their immune systems will be wrong. They will be compromised heavily. We need our bacteria. So my thought is that these viruses are doing something interesting and useful. Now, how we're going to find that out is really not that obvious. But, the kind of thing that came out of this is we're able to actually make a tree of, the hosts and then the phages. again, I'm not gonna go on any further, but you know, that's an example of stuff we're working on. So I hope you guys have a fun additional week. So I'm getting ready to fly home, which is certainly a brutal 30-hour event, but I'll be thinking of you, and it was really fun to be here. So thanks.
Say, on RBI,
Yeah.
so Bonnie thought it was named after the British physiologist.
Really?
But Newton Harvey,
Yeah.
there's a lineage that I think is fascinating.
What is it?
Well, Newton Harvey worked at Princeton. He was a chairman a long time ago.
Yeah.
Isolated vibrio. He trained,
Huh.
Oh, this is just a bit of history about
Ah, cool.
GFP history.
Oh, good.
And it's ethno or paracentric or something like that. So those of you who don't know, I'll say I'm from Princeton, and Harvey-I is named after, Vibrio Harvey-I is named after E. Newton Harvey. And so there's this interesting lineage that you have in science. And he was one of the founding members of the biology department at Princeton, and he trained, the name will go now, the fellow who isolated luciferase from fireflies and was an academy member. He also trained the first guy that worked with Harvey-I and did the precursors of quorum sensing, Woody Hastings at Harvard. And he then trained, and then the next step down in the lineage was Frank Johnson at Princeton, who with Shimomura isolated GFP and purified it. And I think, I'm not so sure Bonnie's the next, you know, like a great-great-grandchild in this tradition, but it's been very strong in this sort of obscure place. It's really quite fascinating.
Yeah. Well, I also think it's just, it's worthwhile for everybody to contemplate, you know, how GFP came onto the scene, you know. So, you know, it's just like, the reason it's worth thinking about is because, at least in the US, we have study sections, learned 20 people sit in a room and decide what other people should work on. And, you know, most of the cool stuff, a lot of the cool stuff isn't figured out that way. Let's say it that way, I suppose. Sorry to be so obnoxious.
So one comment about the viruses and bacteria which lives in our own body.
Yeah.
So Dr. Marshall, he generally works in the immunological disorders, and he figured out that our immune system and whole bodies consist of so many bacteria and viruses, and he figured out that generally there is always a competition between our ecosystem and their ecosystem. And then he figured out that actually our innate immunity is related to that ecosystem of the viruses and bacteria. And sometimes what happens, there is one vitamin D receptors in our cells. So that bacteria and viruses, they take the vitamin D cells receptors from the cells, and that causes some kinds of cancers and some kinds of autoimmunity.
Yeah, cool, thank you. Yeah, so maybe that's going to be...
Diluted salts connected by an ion channel. Is there any way to find out the chemical potential difference between the two salts?
Well, you know, the first thing that comes to mind is that the current is going to be proportional to the difference in the chemical potential, but there's a transport coefficient in there that's probably unknown. So it's not obvious to me how to make that connection. Maybe people that are smarter will figure that out, but I don't, off the top of my head, I don't know. Yeah, no, that's what I'm saying, experimentally. It seems a little tricky, but maybe there's something you can do. I don't know. Okay, look, you can also, just by measuring well, in the cellular context, it seems tricky. I don't know. I'm out of my depth on that one, so I'm not even going to try and answer it. Sorry. Other question? Yeah. Hi.
Thanks for a very wonderful lecture.
Thanks.
I just wanted to ask. I think you were basically talking about how mathematical modeling may reveal underlying similarities between apparently disparate processes.
Yeah.
Now, one question that has been sort of bugging me for quite some time is that we know that at different levels there are regulatory mechanisms. For example, let's say translation and transcription, and cell division, and so on. So is it reasonable to expect that they can be conceptualized as emergent phenomena which arise out of interaction of multiple, more elementary factors and operations? That's question number one. And question number two is that if that might be reasonable, then what kind of, mathematical dynamical systems framework may help us to sort of capture this kind of, higher level regulatory phenomena?
Yeah, so were you there yesterday at NCBS?
No. Yes.
Okay, so Boris Schraman back there, I thought made a very interesting point because he talked about theory with a capital T and theory with a lowercase t. And for me, in general, I kind of focus on theory with a little t, which means take a little tiny problemlet and mess around with it, and one suffers already a lot when you try to do that. Of course, I do think that there are coarse-grained descriptions, higher level descriptions. That's why I was saying what I was about kinetic theory versus thermodynamics. So, but what it's going to look like, one of my favorite examples is the theory of active matter. Active matter. People like Jacques Pro, Ramaswamy, there's a bunch of people that have worked on that. That's a very interesting idea. It basically sweeps the existence of myosin under the rug, and it treats it in a sort of phenomenological way. In the same way that, when you talk about diffusion, that's really the motion of atoms, right? So if you wanted to, you could have an atomic worldview, but you just bury all of your ignorance in D, the diffusion constant. Or when you do elasticity, if I do elasticity of aluminum, there's 3 numbers in GPa units, and that's it, C11, C12, and C44. I don't care about atoms anymore. So, that's the hope. But I would also, what I'm a little worried about, and what we talked about before our little theory thing yesterday, is this course has been kind of oriented towards what we like and what we do and what's worked and stuff. But we shouldn't forget that this is incredibly hard. Most of the time it fails. Most of the time you don't know what the hell you're doing. Most experiments are, like I showed inconsistencies between experiments. It's a mess. And to me, that's actually what science is like, as best I can tell at age 53. Most of the time when you're working on something cool and exciting, it's confusing. So I don't know. I don't have any big, cosmic insights, unfortunately. But I like the active matter. I like elasticity. I like hydrodynamics. And I love thermodynamics. So to me, that's the analogy. For me, every time somebody says, but what about the mechanism? What I'm thinking to myself is, I don't want to have kinetic theory right now. I want to have thermodynamics. It's a little contrarian, but that's I don't know, blah, blah.
Yeah. You showed a slide with a lot of formulas, and you said you can go back and check yourself. Can you tell us where to?
What was it?
There was this MWC model associated with that. There were,
Yeah. What was it?
Maybe the reference for the MWC.
It's a figure in there.
Fine.
It's a great, if you want to suffer for a weekend, go for it. And you can mail me. It'd be fun to chat about. But I think they're right. More than one of us checked them. But some of them are painful. There's also a beautiful paper by Peter Swain, which is in PLOS something or other, from 2 or 3 years ago. And it's really nice. It's got 8 separate features of MWC molecules: dynamic range, leakiness, slope at the midpoint, all that stuff. And he talks about their interdependence. So I'd start there, to be honest. Peter Swain, S-W-A-I-N. Yeah. Yes, sir.
Put the viruses on the viri on the phylogeny. What did you use?
So it's really what you would think of, which is that you use the sequence of the terminase gene, which is the thing that we amplified, and you do sequence comparisons. So it's a tree based on sequence. That's what we think. Ultimately, we don't know. So Ted's question is, were those viruses actually integrated into the bacterial genome, or were they going along for the ride, attached as free viruses? And we think they're lysogens, and the reason we think that is that we determined the primers, or Arbel did, based on metagenomes. And we can go look, a couple of the organisms have been sequenced, and there are prophages inside of them that harbor the terminase sequence. So this is a really interesting thing about viruses. And HIV is maybe the one that all of you would know about, which is that some viruses incorporate their genome into the host. And some of these bacterial viruses do just that. That's called a lysogen. Anything else? Yeah. Okay, thank you.